So welcome to the lecture on organization of epithelial tissues. I'm hoping that at the end of this lecture, there are things that will be very familiar to you now, that you'll have understood much about epithelial tissues. The following are the things you're going to learn in this lecture. First, perhaps just need to define what is epithelium and state the general functions. We'll also look at the key characteristics of epithelial tissues. We'll then look at the concept that we use in classifying epithelium. After that, we'll look at the different types of epithelium, and we'll state the distribution of each epithelial type. And perhaps that's where we'll spend more time. And also perhaps the most challenging part, even from examination perspective, when we give you different epithelial types and we expect you to identify them. That's a part that gives trouble. So we'll spend more time there. Then we look at the apical, lateral, and basal surface specializations of epithelial tissue and state the role of each, and we'll finish with glandular epithelium. So in these other parts, which are straightforward, I think you'll allow me to go fast. Then on the other parts, we can take our time. Let's now address the first agenda, which is to define what is epithelium and state the general functions of epithelium. By definition, epithelium refers to the layer of cells that cover body surfaces. Having said so, what are the general functions of epithelial tissues? They tend to protect the underlying body tissues from physical injury and infections. In some surfaces, like in the GIT, they are responsible for absorption of substances. In some places, like glandular epithelium, secrete substances. In some surfaces, like skin and olfactory epithelium, they detect environmental stimuli, so they're sensory surfaces. And in other surfaces, they promote transport, especially epithelium that is ciliated, like in the fallopian tube or in the respiratory tree. They promote transport of substance through the ciliary activity. So that's the first agenda we're done. The second agenda is to talk about the properties or characteristics of epithelium. The following are the characteristics of epithelial tissues. Fundamentally, epithelial tissues consist of cells which are very closely packed. They are very contiguous, as you can see here. And if the cells are closely packed, then it means this, that the material, the matrix of that tissue is very minimal. We can actually say hardly any matrix. The third property of epithelium is that they lack blood vessels. So they rely on transfer of nutrients through diffusion from the underlying connective tissues. Fourth characteristic is that epithelium rests on what we call a basal lamina, which separates the epithelial tissue from the underlying connective tissue. Fifth characteristic is that epithelial tissues display polarity. By polarity, it means that there is directionality of function. Let me use this one to demonstrate that. If this epithelium was going to absorb substances, it will move from here, the apical surface, to that side, the basal surface. There's some directionality in terms of that. If this epithelium is going to secrete substance, it will move from this side into that direction. So that directionality of function is what we are referring to as polarity. However, from histologic perspective, from a morphological perspective, of course, you can't see the directionality. So what do we see? What we see is that the epithelium is resting on a surface, and by it resting on a surface, there's a part of the epithelium that is attached. We call that the basal surface. And there's a part of it that is free. We call that the apical surface. Lastly, epithelial tissues have a high regenerative potential. And them having a high regenerative potential means that they're actively mitotic, which means that they divide a lot. They're actively mitotic. Right. So those are the general characteristics of epithelial tissues. Now I want us to look at the concepts that we use when you're classifying epithelial tissues. We can classify epithelial tissues first based on function. According to this, we classify epithelium into two types. There's what we call lining epithelium, and there's what we call glandular epithelium. The lining epithelium is the one that is actually on the surfaces of the body, offering protection. The glandular epithelium is the one that forms glands, therefore provides secretions. The rest of the classification of epithelium is largely based on us now breaking down the lining epithelium as opposed to us breaking down the glandular epithelium. And so how do we then classify the lining epithelium? One of the parameters that we use in classifying lining epithelium is the number of cell layers that that epithelium has. On this note, there are some terminologies that we are going to use. It can be simple, stratified, or pseudostratified, and I'll explain. The second parameter that we check after looking at the number of cell layers is the shape of the cells on top. And again, we are going to use some three terminologies: squamous, cuboidal, and columnar. And we'll explain what those are. In most cases, the story will end there. So you look at lining epithelium first, number of cell layers, followed by the shape of the cells on top. But there are times we go to the third parameter, where we check the type of surface adaptation. So on this one, we want to say whether the epithelium has cilia, then we say it's ciliated. Whether it has microvilli, whether it has stereocilia, or whether it's keratinized. We comment on that. Let me say that not all the time we have to look at epithelium based on that third parameter. Most of the time we look at lining epithelium based on the first and the second parameter. But there are a few occasions when we'll have to look at epithelium based on the third parameter. Now let's break down what we've just said. We've said according to function, we classify epithelium into two. We have the lining epithelium, which actually covers surfaces, offering a protective role. And glandular epithelium, the one that provides secretions. And we've said that the rest of the classification of epithelium is now based on us breaking down the lining epithelium. So we classify lining epithelium first based on the number of cell layers. If it's one layer, we call it simple. If it is more than one layer, we call it stratified. We call it pseudostratified if it is one layer, yes, but it's a mixture of short and tall cells. So the appearance it gives us is that of stratification, yet it is not. We call that pseudostratified. So simple epithelium is one layer of cells, everything resting on the basal lamina. Stratified epithelium, when you have more than one layer of cells. So that means that there are cells which will not rest on the basal lamina, these ones. Only the basal cells rest on the basal lamina. Pseudostratified epithelium, everything rests on the basal lamina, but it's a mixture of short and tall cells. So because a mixture of short and tall cells, the nuclear layers or nuclear levels will be different. It gives you appearance of stratification. Based on the shape of the cells on top, squamous means flat. Cuboidal means that the height and the width are the same or near the same. And columnar means that the cells are taller than their wide. Having agreed on the concept that we use in classifying epithelium, let's now give names to different epithelial types. As we do this, we'll also state where these epithelial types are found. So the first one looks like this. I would wish that we have a discussion on it, but let's just continue so that we save on time. Here, we are seeing one layer of flat cells, and all the cells rest on the basal lamina. So you want to call this one simple squamous epithelium. It means one layer of flat cells. Simple for one layer and squamous for flat. Simple squamous epithelium is found in a number of places like the alveoli of the lungs, endothelium, this is the inner lining of blood vessels. We find the endocardium, inner lining of the heart. Mesothelium, this is the epithelium that form the membranes that cover body cavities. So there are three: pleura, peritoneum, and pericardium. The Bowman's capsule is the kidney. The inner epithelium of the cornea is called corneal endothelium. That epithelium may be simple squamous or may be simple cuboidal. Then we have some segments of the nephron, especially the thin segments of the loop of Handel. Those ones are lined by simple squamous epithelium. That is the distribution of simple squamous epithelium in the body. Second type of epithelium would look like this. So we see one layer of cuboidal cells, we call it simple cuboidal. Simple cuboidal epithelium. Okay, sorry about that. Simple cuboidal epithelium is found in some parts of the nephron again, like maybe the proximal and the distal convoluted tubule, or the straight tubules, which could be the thick descending limb or the thick ascending limb. You may also find in some parts of the collecting ductules and the proximal parts of the collecting ducts themselves. We also find it in glandular epithelium. This is the epithelium that forms glands. So basically, that is not necessarily lining epithelium, but most glandular epithelium will have simple cuboidal epithelium. We find it in the epithelium of the cornea as well. Remember, I told you that corneal endothelium, the epithelium on the inside of the cornea, can either be simple squamous or simple cuboidal. Some ducts of the exocrine glands will have this epithelium, especially the smaller ducts. And the thyroid gland also has this kind of epithelium. simple cuboidal epithelium. Third type of epithelium looks like this. One layer of tall cells, so we call it simple columnar epithelium. We find it in some parts of the female reproductive tract. We can name them: fallopian tube, uterus, and remember that one is the one we call endometrium. And the upper part of the uterine cervix, that is what we call the endocervix. So these three parts of the female tract are lined by simple columnar epithelium: fallopian tube, endometrium, and endocervix. Some segments of the alimentary canal are also lined by simple columnar epithelium. If you want to know the segments of the alimentary canal lined by simple columnar epithelium, is straightforward. Just take the journey followed by food, but start from the stomach, go sequentially up to the rectum. So stomach, duodenum, jejunum, ileum, cecum. We can mention the appendix there, although food does not go to the appendix, but it's still lined by simple columnar epithelium. Then the whole colon, and finally the rectum, simple columnar epithelium. Some parts of the renal tubules are also lined by simple columnar epithelium. Same places which were lined by simple cuboidal may also be lined by simple columnar. So the convoluted tubules, whether proximal or distal, may be simple columnar or simple cuboidal. The straight tubules, whether as thick ascending limb or thin ascending limb of the nephron, they're lined by simple cuboidal or columnar. The distal portions of the collecting ducts are also lined by simple columnar epithelium. And some ducts of exocrine glands, especially the larger ducts now, are also lined by simple columnar epithelium. Fourth type of epithelium would look like this. This is what we'll call stratified columnar epithelium. We can see the stratification, and we've seen that the apical cells are flat. Sorry, the apical cells are columnar. Stratified columnar epithelium is found largely in the conjunctiva. And I believe we all know where the conjunctiva is. We may also find it in some ducts of exocrine glands, especially the larger ducts. So larger ducts of exocrine glands have stratified columnar epithelium. Okay, this other one, I want us to look at it again. So we see that this epithelium is by all means stratified. And you may wonder, should you call it stratified cuboidal because of this? Or what should you call it? Importantly, you have to remember what we agreed. You classify epithelium first based on the number of cell layers. So we agreed this is actually stratified. Then the second parameter is to check the shape of the cells on top. So don't be tempted to look at these ones, even though they look more obvious, and now call these ones stratified cuboidal epithelium. That will not be correct, because you can see this is the basal lamina. So this is the basal zone of this epithelium. That means this is the apical zone of this epithelium. The apical cells, as we can see here, are flattened. And so the correct naming for this epithelium would be stratified squamous epithelium. It means we have several layers of cells stratified, and that the cells on top are flattened. We find stratified squamous epithelium in surfaces that are subjected to a lot of friction. For that reason, there are more modifications that we can have on the stratified squamous epithelium. Accordingly, there are 3 subtypes of stratified squamous epithelium based on the degree of friction that that epithelium is subjected to, whether actualized or anticipated. So I want to teach you the 3 varieties of stratified squamous epithelium. We classify the stratified squamous epithelium into 3 varieties based on the degree of keratinization. The first one is what we call stratified squamous keratinized epithelium. This epithelium is largely found on the epidermis of the skin. What do we notice about it, or why do we call it stratified squamous epithelium? It is largely based on the presence of this thick layer here, which is the keratin zone. If you look at this epithelium, you notice that these are cells, yes, several layers of cells, so it is stratified. The cells on top, which are these ones, appear flattened, so squamous. But beyond the cellular zone, we see this region that is acellular. This acellular material is what we are calling keratin. This keratin layer is so thick, as we can see, almost the size of the cellular layer. This is how stratified squamous keratinized epithelium looks like, and this is exclusively found on the skin. Now, this epithelium is adapted to withstand friction based on some parameters. One is the stratification. The number of layers of cells in stratified squamous epithelium is usually unbelievable. You may have even over 10 cell layers. So different layers of cells, stratified. Then keratinization. There's a thick keratin layer that is on top of the cells, helping to withstand friction. And third, the intercellular junctions. You may have already done cell biology, and so you are familiar with the intercellular adhesion molecules. You must have come across what we call desmosomes. Adhering junctions that hold adjacent cells together. So these cells have several desmosomes between them, helping them to be held together. So that is stratified squamous keratinized epithelium found in the epidermis of the skin. The second subtype is this one that we call stratified squamous parakeratinized epithelium. So again, we see stratification. Look at the number of cell layers, many of them. The apical cells are flattened, so squamous. Then we see this material here, amorphous material there. That is keratin. The difference between this one and the previous one is that this one has a thin layer of keratin. We call this one stratified squamous parakeratinized epithelium. That keratin layer that is thin may sometimes even be patchy, which means that some parts may not actually have the keratin lining. This epithelium is unique to the oral cavity, and it's not all the parts of the oral cavity. It is unique to the parts of the oral cavity that comes to contact with food when you're chewing. For that reason, we call this epithelium masticatory epithelium. Take some 10 seconds and think through that. Which parts of the oral cavity actually come to contact with food when you're chewing? So not all the parts, only the parts that are subjected to the friction of food during mastication. Okay. The third subtype of stratified squamous epithelium would look like this. What do we see? We see epithelium that is stratified, and so we're okay there. And that the apical cells are flattened, squamous, so we're okay there. And we don't see any keratin lining on top of that. This is stratified squamous, non-keratinized epithelium. Stratified squamous, non-keratinized epithelium will be present in some parts of the digestive system, like the part of the mouth that do not come to contact with food when you're chewing. Again, think about those ones. The esophagus itself and the anal tube itself. Stratified squamous, non-keratinized epithelium. We agree that either way, those surfaces are subject to friction. Some parts of the reproductive system also have this epithelium. In the female system, the vaginal canal has this epithelium. The lower part of the cervix, which we call the ectocervix, also has this epithelium. The distal aspect of the male urethra also has this epithelium. You agree with me that stratified squamous epithelium is found in regions that are subjected to friction, whether actualized or anticipated. The cornea is also lined by this epithelium. But which side of the cornea? The side of the cornea in front, now not the one behind. That is what we call corneal epithelium, as opposed to corneal endothelium. The corneal endothelium is the epithelium behind. The corneal epithelium is the epithelium in front of the cornea. And then we have the vocal cords. They're also lined by this kind of epithelium. Again, you agree with me that it's subject to friction. Right. Another entity, having moved from stratified squamous epithelium and the three subtypes. Another entity is this one. When you look at this epithelium, we notice that it appears like it is stratified. But a careful look, and perhaps the drawing is the one that will help us. We note that all the cells are resting on the basal lamina. So it's actually one layer of cells, but it appears like there are multiple layers. It is pseudostratified epithelium. When you have pseudostratified epithelium, definitely only the taller cells will reach the top. And so as a concept, pseudostratified epithelium will be columnar. So we call this one pseudostratified columnar epithelium. A mixture of short and tall cells. This epithelium would look like this. If you were to draw it, that's what it looks like. All the cells rest on basal lamina, but you have a mixture of short and tall cells, sometimes even intermediate size. Where do you find this epithelium? There are three subtypes of pseudostratified columnar epithelium. Again, based on now the apical specialization. Remember, I told you that there are some instances where we will require us to classify epithelium based on the third parameter, the structure on the apical surface. So based on this, there are three subtypes of pseudostratified columnar epithelium. The first one looks like this. It is pseudostratified, ciliated. It has cilia. This is the cilia. But in addition to that, we see some cells here, like that one, or that, which do not appear to take the stain. They don't take the stain because they have mucus. So we call them goblet cells. They are mucus cells. This epithelium is called pseudostratified columnar ciliated epithelium with goblet cells. This epithelium is unique to the lining of the major airways. So nasal cavity, some parts of the pharynx like nasopharynx, the larynx, the trachea, and the bronchi. Those are major airways. They're lined by this epithelium. Because this epithelium is unique to the respiratory tree, we also call it respiratory epithelium. That's the first variant of pseudostratified columnar epithelium. The second subtype of pseudostratified columnar epithelium is pseudostratified columnar ciliated epithelium without goblet cells. So this is pseudostratified columnar again, and this is the cilia, but we don't see goblet cells here. This epithelium is unique to the upper part of the nasal cavity. Remember, the upper part of the nasal cavity houses the receptor for smell. So this is actually the epithelium that functions as the receptor for smell. For that reason, we call it the olfactory epithelium. That's the second subtype of pseudostratified columnar epithelium. The third subtype of pseudostratified columnar epithelium is this one, which we call pseudostratified columnar epithelium with stereocilia. Perhaps let me use this chance to also teach you how do you pick from light microscopy that this epithelium is actually stratified and not pseudostratified, or vice versa. How do you know this epithelium is pseudostratified and not stratified? At least the simple one we can pick directly, because we'll just see like almost one nuclear zone. Then you know that the cells are of the same height and same level, because you see relatively one nuclear zone. Now, look at this one. We see different nuclear zones. Some nuclear there, some nuclear there. Remember, this is like a lumen. And so this is the apical surface. This is the basal surface of that epithelium. So we are seeing different or multiple nuclear zones, which means that definitely it is not a simple epithelium. So maybe it is stratified, maybe it is pseudostratified. How do you distinguish the two? This is what I want you to do. Imagine that you are able to divide this epithelium from the basal side to the apical side into zones. The number of zones you want to divide it to doesn't matter. Maybe divide it into 2 zones, maybe 3, maybe 4. I usually prefer 3 or 4. So let's say we're to divide it into 4. That would be zone 1, zone 2, zone 3, and zone 4 there. So that's 4, 3, 2, 1. Now, the numbering doesn't matter. You can still call this one zone 1. The number is not conventional. And remember, this is in your mind. How do you distinguish stratified from pseudostratified? In stratified epithelium, the nuclei will be present in all the zones. While in pseudostratified epithelium, the apical zones, these ones, will be relatively lacking the nuclei. They are relatively devoid of the nuclei. And this one is so classical. We see several nuclei at the basal zones, but we see hardly any nucleus on the apical zone. That is how pseudostratified epithelia look like. So I've used this to help you understand how to pick pseudostratification on light microscopy. Anyway, now narrow down to this that we are calling pseudostratified columnar epithelium with stereocilia. This epithelium is pseudostratified columnar, yes. We also agreed that if you have pseudostratified columnar, then definitely it is so when you have pseudostratified epithelium, then definitely it's columnar. As a concept, you have a mixture of short and tall cells. So which ones will reach the top? Only the tall ones. So here, look at these things which extend into the lumen. These are the ones we call stereocilia. We'll be talking shortly about them. So this epithelium is pseudostratified columnar epithelium with stereocilia, very unique to the male genital duct system, the path followed by sperms from the testes to the exterior. Now, not all the parts of the ductal system of male will have this epithelium. We can name the specific types. From the testes, sperms go to epididymis. Epididymis have this epithelium. Actually, this is epididymis. After epididymis, sperms go to the vas deferens. And after vas deferens, sperms go to the ejaculatory duct. We can put those three as having this epithelium. After ejaculatory duct, sperms go to the urethra. Urethra has a different epithelium. What is the role of stereocilia in this epithelium? When sperms are produced from the testes, to facilitate the movement of sperm from the testes to the epididymis, the site of storage and the site of decapacitation, we need some fluid which are produced by the Sertoli cells. So the secretions of the Sertoli cells help to move the sperms from the site of production, testes, to the site of storage, epididymis and the vas. Once they have reached the site of storage, that fluid is absorbed back. Otherwise, it will swell a lot. So the role of stereocilia is to absorb the fluid that has been produced by the Sertoli cells that push the sperms to the epididymis and vas. Some people want to call this epithelium germinal epithelium, although that may be a misnomer, because there's also another germinal epithelium that covers the ovary, and it's usually simple cuboidal epithelium. So I discourage you from using the term germinal epithelium. I've just mentioned it because some books may have it. Let's look at this one. Very unique. There's some things I want you to capture in this image. One, we understand or we can appreciate that the epithelium is definitely stratified. When you look at the shape of the cells on top, we can also appreciate that the cells on top are actually cuboidal. So we can say this epithelium is stratified cuboidal, and there's nothing wrong with that one. I want you to look at the apical cells carefully. And you'll notice that there's some cells have two nuclei instead of one. Have that in your memory. So there's some cells which are binucleated, even though most of them are uninucleated or mononucleated. A few apical cells are binucleated. Now look at the shape of the apical cells. They look like they bump into the lumen, somewhat dome-shaped, or rather rounded. They look like the top of a mushroom or the top of an umbrella. These cells are very unique. When the epithelium is not stretched, which means relaxed, the apical cells are more rounded. And the number of cells are even many. In this image, we are seeing how many? 1, 2, 3, 4, 5, roughly 5 cell layers in this one. And in this one, we are seeing how many? 1, 2, 3, also 5. It doesn't have to be 5. Coincidentally, the two drawings said 5. So when the epithelium is relaxed, the apical cells are rounded. They have a rounded contour. And the number of cell layers are more. So we're using the term 5 for more. How about when the epithelium is stretched? This is how the epithelium will look like when it is stretched. First, the number of cell layers have reduced. They are no longer 5. They are now 4. So some reduction we've seen. Apart from that reduction, we also appreciate that the cells on top now have a slightly flattened outline. They are moving towards being squamous. This type of epithelium, therefore, changes a lot. On two perspectives you've seen. The number of cell layers change. Sometimes the number are more, sometimes the numbers are less, depending on whether the epithelium is not stretched or stretched. Then the second parameter that changes is the shape of the cells on top. Originally, they were cuboidal, but now they are moving towards being squamous. So how should we call this epithelium? We call it transition epithelium. So the naming is based on the fact that some parameters of the epithelium change. The number of cell layers change. The shape of the cells on top also change. Transition epithelium is the epithelial lining of the urinary passages. When there's a lot of urine less in the bladder, the epithelium is stretched, it will be having fewer cell layers, and the cells on top will have a flattened outline. And when there's no urine, the epithelium is relaxed, there'll be more cell layers, and the cells on top will be more cuboidal. So we call it transitional epithelium. We find it in the following sites: minor calyces, major calyces, renal pelvis, ureter, urinary bladder, and the urethra. So those are the urinary passages. Because it is unique to the urinary system, we call this epithelium urothelium. So that when you talk of urothelium, we know you're referring to transitional epithelium. Let's look at this last one. Very unique as well. Perhaps before we give it a name, I give you a chance to just write on the chart how you do classify this epithelium. I've made the chart in such a way that I'm the only one able to see what you're going to write. So feel free and write something. Just identify the epithelium. Let me call this one question 1 to help me distinguish what you've written so far. Look at this epithelium again. So we agree that yes, the epithelium is of a stratified type. And most of you alluded to that. But look at the shape of the cells on top. The truth of the matter is that you cannot really commit to any particular shape. So those ones are cuboidal, yes. How about these filamentous ones? Which shape are those ones? Because we cannot commit to a particular shape, the shape of the apical cells are largely indeterminate. So we can't use the shape of the cells to classify this epithelium. And I think that's where most of you went wrong. But there's something else I want you to see. There's some filamentous projections into the lumen. Many of you have been deceived to think that this is cilia. Cilia is not that long. There's some filamentous projections. This epithelium is termed complex stratified epithelium. So you understand why stratified? Complex because we cannot really tell the shape of the cells on top. No particular shape that we can commit to. We call it complex stratified just for simplicity of grammar. We wouldn't have called it stratified complex. Complex stratified epithelium is the epithelial lining of the seminiferous tubules of the testis. And so these filamentous things you're seeing are actually long tails of the sperms. They are not cilia. Complex stratified epithelium is therefore also known as seminiferous epithelium. Great. So those are the different types of epithelium. Okay, this one is a misplaced one, but still showing us complex stratified epithelium. Now we are done with that long agenda, but I'll want to give you some practice questions on that one shortly. Before we do that, let's look at the 5th agenda. We look at the specializations which are present on epithelium. We'll be quick on this one. So epithelium has some surfaces. The apical surface is the surface on top. We've already clarified that. The free surface. The basal surface is the surface down here that is attached to the basal lamina. Sometimes you may hear these terminologies interchangeably, basal lamina and basement membrane. They are not quite the same. The basement membrane usually has 2 zones, the basal lamina and the reticular lamina. Reticular lamina is largely secreted by the connective tissue here, and the basal lamina is largely secreted by the epithelial tissue, and the two constitute the basement membrane. But in terms of describing epithelium, even if you say epithelium rests on basal lamina, it's still okay. Or rests on basement membrane, it's still okay. But you now know there are 2 different entities. Okay, one is part of another one. So this is the basal surface of the epithelium. This is the apical surface of the epithelium. But there's also this surface where the cells are adjacent to one another. That is the lateral surface of the epithelium. Having said so, epithelium displays some adaptations on the three surfaces. Let's start with the adaptations on the apical surface. So apical surface will see some adaptations in form of projections. We're going to see them. Lateral surface will also see the adaptations there. And basal surface will see the adaptations there. Adaptations on the apical surface of the epithelium include microvilli. Microvilli are finger-like projections from the apical cell membrane. I'm rushing through this because this is particularly part of cell biology. And so most likely you've talked about it. So that's why I am rushing through. Microvilli are usually anchored by actin filaments, which are microfilaments. The aim is to increase the surface area over which absorption takes place. So therefore, we find it in highly absorptive surfaces, like the small intestines, renal tubules, specifically the proximal convoluted tubule of the kidney, and in the ducts of salivary glands. How about stereocilia? Stereocilia is just elongated microvilli, so structurally similar, although stereocilia tend to exist in groups, as we can see here. Two key functions for stereocilia. One is absorption, just like microvilli. And that's the role they play within the male reproductive tract, the epididymis and the vas deferens. But stereocilia is also important for mechanoreception. And that's the role it plays in the ear. These are the ones that function as mechanoreceptors to detect the changes in head position in the ear. The third one is cilia. Cilia are filamentous projections from the apical surface, as you can see. And uniquely, cilia is anchored by microtubules. Also, cilia is able to beat in a particular rhythm. The rhythmic beating of cilia propels substances on the surface of epithelium to one particular direction. Occasionally, we may ask you to state two differences between cilia and microvilli. So remember, in terms of the cytoskeleton that anchors cilia, it is microtubule. The cytoskeleton that anchor microvilli is actin filaments. And then remember, cilia is motile, while microvilli is not motile. So those conditions that affect microtubules may affect cilia, but will not affect microvilli. This shows you cilia at high magnification. And that also, we are seeing cilia as well. Although it's a drawing, but we can see cilia there. In this particular one, mimicking the respiratory epithelium, so pseudostratified columnar ciliated epithelium with goblet cells. The role of the cilia here is to propel the mucus on it. It forms part of what we call the mucociliary rejection current of the airways. These are collaboration between the mucus and the cilia. And remember, the current is usually towards your throat. Then depending on your habit, you may either swallow or spit. We also have cilia along the fallopian tube, and that helps to move the oocyte from the site, from the ampullary end of the fallopian tube towards the uterine end of the fallopian tube. So if you have a disease that affects ciliary activity in terms of the rhythm, the movement, in the respiratory tree, you'll be subjecting this person to recurrent respiratory tract infections. And in the fallopian tube, perhaps you'll expect some level of infertility or ectopic pregnancies. The fourth apical adaptation is keratin. We've already alluded to keratin when we talked about stratified squamous keratinized epithelium. Keratin is the name given to the intermediate filament that is found in epithelial tissues. I hope you do remember in muscle, you call it desmin. In astrocytes, you call it glial fibrillary proteins and the like. So keratin is the name given to intermediate filaments which are found in epithelial tissue. Now, we call epithelium keratinized if you have a thick layer of keratin on top of the cell layer. It's usually just a dead material there. And so you may have it in stratified squamous keratinized epithelium, like that of the epidermis of the skin, or in the masticator epithelium, stratified squamous parakeratinized epithelium. And I saw some people asking the epithelium of the tongue. Remember, I gave you the concept. The parts of the oral cavity that come to contact with food will be stratified squamous parakeratinized, which you call masticator epithelium. The parts of the oral cavity that do not come to contact with food when you're chewing, stratified squamous non-keratinized epithelium. So ask yourself, which side of your tongue come to contact with food? And which side of the tongue don't come to contact with food? The lower parts don't come to contact with food. That's the ventral aspect of the tongue. So that one is stratified squamous non-keratinized. The top surface of the tongue, the dorsum of the tongue, come to contact with food. So that is masticatory epithelium. So anyway, keratinization is an adaptation to withstand friction. Those are apical surface adaptations. How about lateral surface adaptations? I'll be quick on this because you may have done it under cell biology. So you may have some things that hold adjacent cells together. We have tight junctions. Tight junctions are also called occluding junctions. These ones prevent paracellular transport. They only allow transcellular transport. If this epithelium was to be absorptive, it will not allow substances to move between the cells. It will not allow substances to percolate between the cells. It only allows substances to move through the cells so that the cells can control what is being transported. So we have tight junctions or occluding junctions where we need controlled transport. We can call them zonular occludents. At their junction are desmosomes. So these ones just hold adjacent cells together. There is usually a disc that comes from each cell. So one disc from one cell, another disc from another cell, and that makes a complete disc, which we call desmosome. You can also call them zonular adherents. Then we have the communicating junctions, which are also called gap junctions. These ones allow free ionic movements from one cytoplasm to another. So we also have it in some epithelial cells, although maybe you have it more in cardiac muscle and smooth muscles and perhaps some neurons. The basal surface adaptations are two. One is hemidesmosome. Understand it's just a desmosome, except that now we are not talking about two cells. We are talking about a cell and a basement membrane. So you'll only have one disc instead of two discs. So because you have only one disc instead of two, we then call it hemidesmosome. Then the basement membrane of the basal epithelium, basal side of the epithelium may fold a lot, and this is what we call basal membrane infoldings. Basal membrane infoldings are just mechanisms of increasing the surface area of the basal side of the epithelium. Again, adaptation for absorption. So those are the epithelial adaptations. But before we look at those, the last agenda on glandular epithelium, I want us to recap something that we said, so that I'm going to give you a test where you're able to identify different epithelial types. Give me a second, I upload that one. So this is it. I want to take this opportunity to summarize for you how you practically pick the different types of epithelium when you are given to them under light microscopy. This will help you to understand and also to pass your practical exams in histology as with regard to epithelial tissues. So the moment you look at a slide, what will make you think that this is epithelium and not any other tissue? Well, we know that we have 4 basic tissues: epithelial tissues, connective tissues, muscular tissue, and nervous tissue. What will make you narrow down to the fact that you're looking at epithelial tissue? In light microscopy, you'll be relying on very few things. You know, I want you to understand now from a practical point of view. There are those things we said about, oh, the epithelium is avascular, has high regenerative potential, and the like. Very true, except you won't look at those ones under microscope. So what do you see under microscope? One, the cells are closely packed, which means that the matrix is very minimal. For me, that is very key. That if I don't see cells closely packed, then it is not epithelium. And what next? Is it on a surface? Because that will make me know that it has a free and a basal surface. It's attached onto something. These two are very fundamental. If you don't see those two, most likely you're not looking at epithelium. And you see those two, most likely you are looking at epithelium. So having said so, once you've established that the slide is epithelium, what next? The next thing is to classify the epithelium. What do we check? You first check the number of cells. We will not see the outline of the cell membrane on light microscopy. What you'll see is the nuclear zones. If you have only one nuclear zone, or appear like almost one, then call it simple epithelium. If it's multiple nuclear zones and it appears like the nuclear zones are occupying all the zones of the epithelium, then call it stratified epithelium. If you see multiple nuclear layers or multiple nuclear levels, and that the apical zone is devoid of these nuclear levels, call it pseudostratified. After you've done that, then look at the shape of the cells that adjoin the apical surface. If they are flat, call it squamous. If they are cuboidal, if they are tall, call them columnar. Again, you are not going to see the outline of the cell membrane. So how will you know that this cell is squamous or columnar? Sometimes you may be able to see the outline of the cell membrane, and that would be straightforward. If you don't see, you rely on the shape of the nucleus. If the nucleus is more vertical, then the cell is most likely columnar. If the nucleus is more rounded, then the cell is most likely cuboidal. If the nucleus is more flattened or more horizontal, and we are looking at the plane of the apical surface to judge those terminologies. If the nucleus is more horizontal, then we call it squamous epithelium. The third parameter you check is the type of apical surface adaptation present. And most of the time we'll not look at this, but sometimes you can check it. So is it cilia? Is it keratin? Is it microvilli? Is it stereocilia? Having said so, let's start with this one. I'm calling it question 1 for you. I want you to answer in the chat. Give it a name. Okay, let's do it a different way. Let's just have a discussion about it. So anyone who wants to give me the name to this one, just put up your hand and I'll enable your mic. Yes, Ikambili.
I'd say stratified, squamous, non-keratinized.
Very good. That is stratified, squamous, non-keratinized. Thank you, Ikambili. So this is actually the epithelium from the esophagus. Okay. That's another one. I call it question 2. I think I have 2 magnification levels for this, that level and that level. Okay, I'll give it to Joseph Mbugwa.
This is a keratinized stratified squamous epithelium.
Okay, so that is okay. We can just change the order of in which you've said them. So we said stratified squamous keratinized epithelium. Good, we're doing well. I'll give you a third one. So that is the third one. Any volunteer on this? Any volunteer for this? Yes, Salim Halim. Okay, you dropped again before I could enable your mic. Somebody else? All right, you're now here. Yes, Salim. Carry on, you can increase your volume. Dr. Salim, we can't hear you, so I'll mute you. Let's give it to Anita.
Stratified, cuboidal.
Okay, Anita, you are right, but don't mute fast. Let's look at it again. So I agree with you, it is stratified, and I agree with you that it is cuboidal. But there's something I want you to notice. There and there.
Oh, it has 2 nucleus.
Okay, so the apical cells are binucleated.
Yes.
I also want you to notice the shape of the cells on top. They bump into the lumen like a dome. So what did you call this epithelium now?
like a dome. It's stratified, cuboid, transitional.
Yes, now we call it transitional epithelium. So you don't have to call it stratified cuboidal epithelium. This is specifically urothelium. Thank you, Doc. Okay. Number 4 is this. Any volunteer for this? Yes, Christian.
It's pseudostratified columnar epithelium with goblet cells.
Okay, I want you to look at it again, Doc. So this is the epithelium, that one, and this one. Also this one, and that one. And finally that one. Let's focus on this one. How many cell layers do you see?
One.
Only one. So it is not pseudostratified, because we can see only one layer of nuclear, one nuclear layer. So it's a simple epithelium. Is it simple columnar, simple squamous, or simple cuboidal, Christine?
Simple columnar.
Very good. So this is simple columnar epithelium. And I am happy that you've noted that we have goblet cells there. But just before you mute, what do you see here?
Some keratinization.
Well, it's not keratinization. When you see that double line, very compact double line, we call it brush border. And brush border is how microvilli look like under light microscopy. So this is simple columnar epithelium with microvilli and goblet cells. Okay, thank you, Doc. Simple columnar epithelium with microvilli and goblet cells. And this is actually the epithelium of the small intestines. Okay, let's try another one. Number 5, any volunteer? Yes, Stephen?
So it's pseudostratified columnar.
Okay.
With that's stereocilia.
Very good. So this is pseudostratified columnar epithelium with the stereocilia. Okay. Another one, number 6. Anyone? Let's try today rather than wait for the exam to try it. Yes, Emmanuel.
This is the complex epithelium.
Well, this one is not complex, Doc, because we are able to determine the shape of the cells on top. So it's not complex. Somebody else want to try? Yes, Stephen?
Just trying. It looks like pseudostratified columnar epithelium
Yes,
without goblet cells.
Yes, this is the stratified columnar epithelium without goblet cells. We can see that the apical zone is devoid of the nucleus. So this is pseudostratified columnar. Now, something to guide you as well. When you look deep into the deeper layers, you see these other things layer. These are nerve bundles. So this epithelium has several nerve bundles. It tells you that it's a sensory epithelium. The correct name for this one is olfactory epithelium. Pseudostratified columnar ciliated epithelium. The cilia is not that prominent though. without goblet cells. All right, 7. Any trial for 7? Yes, Oscar. Okay, Oscar, I think you have a question. You feel free to ask.
On the last slide,
Yes.
I wanted to clarify if that looks like a brush border. And also I wanted to ask under microscopy, how would you be able to differentiate between the stereocilia and cilia? Because functionally, you can be able to find out, we've been able to see the function, the absorption, and the anchoring microfilaments, but you cannot be able to see that under microscopy. So how would you be able to differentiate it?
Okay, good question. So now this one, it's not a good one for cilia, I agree with you, but I'll still not call it microvilli. Microvilli, you see 2 parallel lines, like a tram track appearance. Let me show you that shortly. Sorry. This is microvilli. 2 parallel lines, very discrete, very uniform, compact together. That's microvilli. Stereocilia is usually elongated and they tend to appear in groups. So we see some grouping, like that one is a group, that's another group. That is stereocilia, elongated, and they tend to be joining on top so that it look like a pyramid kind of for each group. That is stereocilia, very long. Now cilia, depending on the age of the animal that has been sacrificed to do the slide, it may really vary a lot, but generally it's filamentous. I am not so happy with mine here, but I think you've seen some cilia before, and perhaps you're going to see in the next slide again, a better cilia. This was a slightly older animal, and that's why the cilia there is crumbled. So Oscar, hope that is better for you.
Yes, Doc, thank you very much.
Welcome. Okay, Abner, you have a question as well?
No, I wanted to try question 7.
Oh, okay, let me give it to you. So that's question 7.
I was thinking ciliated columnar with goblet cells, something like the respiratory.
Okay, that's true. So let's just have the terminology right. Pseudostratified columnar, ciliated epithelium with goblet cells, and that is what we call respiratory epithelium. So here I am, thank you, Doc. So here I'm confident about the cilia. We can see the cilia there. We are not seeing that double line discrete that we saw for microvilli, but yes, you still see something. That's how cilia looks like. Why are we saying this is pseudostratified? We can see the apical zone is relatively devoid of the nuclear layers, and we can see that we have the goblet cells here. So pseudostratified columnar, ciliated epithelium with goblet cells. This is respiratory epithelium. Dorothy, do you have a question, or it was on the previous question as well?
I have a question. Sorry, my internet is lagging, so I'm not able to keep up. I see them after they've been raised. But I had a question on number 3 and 6. For instance, I felt like number 3 could have been pseudostratified the same as 6, but 6 was actually put to the urothelia. Sorry to ask this, but like, so I want to tell the difference.
Okay, okay. So we are on number 3 here.
Yes. How exactly did you tell that this is not pseudostratified if 6 was pseudostratified?
Okay, good question. We clarified, except perhaps that was the time your mic also gave you trouble. So we said that you divide the epithelium from the basal side to the apical side into zones. Then look at the distribution of the nuclear layers. If the nuclear layers are in all the zones, like this one, that is stratified. But if the apical zones are relatively devoid of the nuclei, then that is pseudostratified. So here we can see the cells are reaching the apical surface there, and we see nuclear there. All the zones have nuclear. So this is definitely stratified, and Doc had identified the stratified cuboidal, and we clarified that we don't just have to call it stratified cuboidal because of the other things we've seen. Let's call it transitional epithelium. If you are to have an actual pseudostratified, and number 5 is an actual pseudostratified, again, we divide it into zones, and we see that the apical zone, in this case, this one, is relatively devoid of the nuclear layers. So this is pseudostratified. So, Doc, I hope that answers your question of how you distinguish between pseudostratified and stratified. Is that okay, Doc?
Yes, it's okay. I actually missed that explanation. Thank you for
no problem.
Again.
No problem. Okay, we see Francis also with a question. Francois, is that Francis?
Yes, it's the English version.
Okay.
Yes, so my question was on question 6, the question number 6.
Yes, it's here.
Yes, I just have a little word with the answer. I think we agreed it was pseudostratified columnar epithelium with cilia without goblet cells.
True.
Yes. Okay. My question is, if you don't include the without goblet cells, is it wrong?
No, it's not wrong, except that you'll be having 2 options there. Because if you don't tell us whether goblet cells are here or not, we will still be wondering, is it respiratory epithelium or is it olfactory epithelium? The distinction is on the presence or absence of the goblet cells. Is that okay, Francis?
Yes, it's okay.
Good. Okay, let me see if I have another one. I think I do. Okay, this one wasn't meant to be there. Let's keep it. Okay, it's already here. I'll talk about it. So this one is showing us a gland, but at a high magnification, we see that the glandular units are largely made up of simple cuboidal epithelium. This is actually histology of the breast. Maybe you'll talk about glandular epithelium you'll talk about breast histology another day. But this just captures for you simple cuboidal epithelium that form glands. This is the last example I'm giving you. Maybe you don't have to answer. It's a straightforward one, so I can take it up. We are looking at the slide of the cornea. This is low magnification, and that is high magnification. On this magnification, we see 2 epithelial linings. First, we agree they are epithelia because the cells are compacted and they are on the surfaces. On one end, the left-hand side, we see stratified squamous stratified squamous epithelium, which is non-keratinized. So that is stratified squamous non-keratinized epithelium, and that's a corneal epithelium. This other end, we see simple cuboidal epithelium, and that is the corneal endothelium. Right, so let's use the next 10 or so minutes remaining to talk about organization of glandular epithelium. So I told you that we can classify epithelium as either lining or glandular epithelium. The glandular epithelium is the one that forms glands. Lining epithelium is the one that is protective generally. When we talk of glands, there are two types of glands. There are those that we call endocrine glands, and there are those that we call exocrine glands. The exocrine glands produce body fluids into ductal system. Endocrine glands produce hormones into the bloodstream. Now, there are some differences between an endocrine gland and an exocrine gland that we'll expect you to know. However, you will have a lecture on endocrine gland histology sometime later, but you'll not have another lecture on exocrine gland histology. So I'll use this moment to take you through exocrine gland histology. If you are to check the differences between the two, we'd expect the endocrine glands to have a lot of blood vessels anyway, compared to the exocrine glands. I believe we can be able to name examples of exocrine glands and examples of endocrine glands. So let me skip that. So in terms of embryology, this is how glands usually develop. They arise from the epithelial lining, whether it's endodermal or ectodermal or mesodermal, as a down growth. And for the endocrine glands, they detach from the epithelial surface, and then they become surrounded by blood vessels. Those will form endocrine glands. But for exocrine glands, they retain their attachment to the epithelial surface, and that attachment will canalize so that you have a ductal system. And so histologically, an exocrine gland has two parts, the secretory portion and the ductal portion. These images capture that well. We have the secretory portion and the ductal portion. We can classify exocrine glands based on a number of parameters. The first one is based on the structure of the secretory portion. If the secretory portion appears like this, then we call that tubular gland. On histology, this is what you'll see. In cross-section, this is what the microscope will show you. It will be a tubule that has been cut in cross-section. You may be very lucky if you capture it in the longitudinal section, of which it will then be straightforward. But this is what you'll see in 2D most of the time. So that is a tubular end piece, the secretory portion. You may also have what we call a cynomorphology. A cynomorphology is where the secretory unit consists of cells which look like prisms, or let me say pyramids. So they have a broad base and a narrowing apex, and that make the lumen of the secretory portion to be a bit tiny, as we can see this one compared to this one. So this is what we call a cynomorphology. Then the third type, what we call alveolar morphology, is where the lumen of the secretory portion is actually wide enough, and the cells are actually simple cuboidal, definite like that. And so the cavity there is a bit wide. That is alveolar morphology. So based on the structure of the end piece, the secondary portion, we have tubular morphology, acinar morphology, and alveolar morphology. We can also, okay, so this just show you some examples of that. We can also classify exocrine glands based on the morphology of the ductal portion. If the ductal portion is single, it's not branching, then we call it a simple gland. But if the ductal portion is branching, we call it a compound gland. So this would be considered a compound gland. And these three would be considered simple glands because the ductal portions are not branching. Then we can classify them according to the type of secretions that they release. How would you know the type of secretions that a gland releases? This is particularly important for the ones which are of acinar morphology. When you have an acinar gland, you want to look at how the acinar cells are actually staining. So in this first one, let's take this to be the acini. We agree that in the acini, the lumen is very tiny. So we are hardly seeing the lumen, but these are secretory acini. We can see on the first image that the secretory acini are deeply staining. We call that one serous gland. If the secretory acini are deeply staining, it means that the secretions of this acini are water soluble, because the dyes that we use in histology are water soluble dyes. So the secretions are hydrophilic, and that's why they are able to take the dye. So they're deeply staining. So we call those on serous glands. Then you can also have what you call mucus glands. The mucus glands have their acini not staining. They don't take up the stain because they contain mucus. So we call those ones mucus glands. And you can see the secretory acini here are not taking up the stain because they are hydrophobic. So they don't take up the dyes that we use. But you may have a combination of the two. And that's what you call seromucus. So based on the type of secretions, you will have serous glands, mucus glands, and seromucus glands. Lastly, we can classify exocrine glands based on how they release the secretion, the mode of secretion. There are some exocrine glands that only releases the content through the process of exocytosis. So the cell remains intact, only the contents of the secretory vesicles are released. We call those ones merocrine glands. There are some cells, or rather there are some glands which release the contents of the secretions, but a part of the cell also disintegrates. Only a part of the cell disintegrates. We call those ones, when the whole cell disintegrates like this one, we call these ones holocrine glands. Then when a part of the cell, this other one, only a part of the cell is disintegrating, we call that the apocrine gland. So let me say that again. In the first one, the contents are released through exocytosis and the cell is remaining intact. That is merocrine mode of secretion. In holocrine type of secretion, the whole cell disintegrates. And in apocrine type of secretion, a part of the cell disintegrates. So we classify exocrine glands based on those 4 parameters. The type of the secretory portion, you have 3 options: acini, tubular, and alveolar. Based on the structure of the ductal morphology, can be simple or compound. Based on the type of secretion of the acini, mucus, serous, or seromucus. And lastly, based on the mode of the secretion, merocrine, holocrine, and apocrine. So as you study epithelium and glandular epithelium, you'll come across some of them. And when you watch this video or when you get the PowerPoint, you're able to get examples of what is now simple tubular gland, simple tubular glands, simple branch tubular gland. You're able to get simple acina gland, branch acina, compound tubular, tubular acina, and the like. There are many terminologies that we can now use to combine to give those namings of exocrine glands. All right, so these ones, I think you can get in any textbook. And so I don't think I'll belabor in mentioning them or giving examples. So that will be the end of that lecture on epithelial tissue organization. I believe your next lecture will then be on connective tissue biology. So we'll stop there.